Miss Gaston. Well, we just wanted to go over Unit 3. So we're just going to kind of do an overview of this unit. We're probably not going to go through every single chapter. But we just wanted to go over the stuff that either we found hard or we wanted to review. So we're going to go ahead and start with Chapter 7, which is Membrane Structure and Function. I'll start off. So first, the cell membrane, or the plasma membrane, is selectively permeable. So that means it allows some substances in more easily than others, and that is because
it's a amnophilic molecule, which means that it has both a hydrophilic and a hydrophobic region.
Yes, and we have a diagram or a little drawing of this phospholipid bilayer, and Jose's going to show it. But as you can see, you have your hydrophilic head facing water, allowing for absorption in, and you have your hydrophobic tails more inwards because, and you guys already said, or did you not say?
It makes it harder to pass the water to pass through.
Yes, which the outside is the hydrophilic layer,
heads, yes.
and the middle of the layer is hydrophobic, which does prevent many things from going in, which mostly water actually.
Yes.
So I'm gonna go ahead and skip into the transport portion because I feel like that's what we have the most trouble with, I think, understanding. It's probably the most important or key concept. So first, passive transport. Diffusion is the movement of particles of any substance, and it doesn't require, since it's passive transport, it doesn't require any form of energy. So it's so much more easy, so much easier for water or oxygen to pass through these cells.
Right. And so whenever water crosses due to diffusion or facilitated diffusion, it is called osmosis. And what is facilitated diffusion?
Well, facilitated diffusion. I wanted to discuss the movement of water, so we'll go into that a little bit later. But water is always going to move from high concentrations of solution to lower concentrations of solution. And so moving down this method is actually called the concentration gradient. And so it'll never go from a lower concentration to a higher concentration.
Yes, and this is, it has tonicity. So there are three basic parts of tonicity. Isotonic, which is the equilibrium. Hypertonic is when the concentration out of the water outside crack is higher than inside, and then hypo, which is the reverse.
Yeah. So when you have a cell that is isotonic, it's nice and it's healthy. It's getting a good amount. I mean, in animal cells it is, but not in plant cells. Plant cells, it has to be hypotonic because it has to have the truancy. I didn't say that right. Okay.
One way to actually explain it is isotonic usually has an equal amount of flow of the solution going through both sides. It's not really still, but instead the solution is flowing through both sides. And the hypotonic, it is filled with of the solution, so it busts. It's more concentrated on the inside rather than the outside, so it combusts. And hypertonic is the opposite. It has less than the outside, so it's I don't know how to show it. It shrivels up, and basically there's kind of a picture of it. It's kind of a little bad if you can't see it, but this is isotonic because the flow of both is an equal amount, and the other's the other way around.
Right.
Yeah.
Which leads us into facilitated diffusion, which is passive transport with the help of proteins, but not ATP because it is still
Because that would be active transport.
So there's two types of facilitated diffusion, which are
cytoplasm and carrier protein.
Well, I think what Adam was trying to say, it's channel.
the
There's
channel ones.
Channel ones, yes. there is gated channel ones. They open. I think the reason they're called gated is because they open like a gate, which is an example of this would be the sodium potassium pump, which
But it uses ATP.
yes, my bad.
So it would be
That'd
an
be an
active,
active, but it-
transport.
But it still uses the gate.
The gated.
So both passive and active can use the gate one, but the channel protein is only to passive transport.
The protein pump, it uses an ATP to open up the pump, and then it takes it in and then opens up again. It's hard to describe it. It's more of a picture. I don't know. Hold up. You should tell me.
Try it out for us, Jose, if you can explain it.
So the cool thing about the sodium potassium pump and the protein pump is that the sodium potassium pump is only in animal cells, but the protein pump is in plants, fungi, and bacteria. And both of these pumps are electrogenic pumps, which means they have a voltage across their membrane. So these are just the different types that are in the plants versus the animals in those different types of cells.
Okay, I want to make sure that I feel like I wasn't clear enough earlier, but in the passive transport, there are 3 different types. I don't think I was clear enough. There's just the diffusion, which is far slower of a process because it has to go through the bilayer. But and then the other the facilitated diffusions are the ion gated and the channel.
Mhm.
This channel is more of a straight, like more thorough. That's what osmosis is really.
More flow.
Yeah, it's more of a flow. True, that's a good word for it.
Yeah, and they're hydrophilic.
Yes.
So it makes it easier for the water to
Which-
pass through.
Which makes sense why osmosis would be.
Mhm.
And I think we can go into aftershock. What kind of went over?
How about exocytosis?
We can go into
Exocytosis and endocytosis?
yeah, exo and endocytosis, which are like, so we have the electrogenic pumps, and then you also have your exo and endocytosis. So
Exocytosis has only one form. It's really just outside the cell. But endo, it's because it's inside, I guess, the inner workings. It has 3 different forms. It's phagocytosis, pinicocytosis. I don't even know how to say them. And receptor-mediated endocytosis. This one, the one that tripped us up in class, the phagocytosis.
Oh my gosh.
It like engulfs other portions, like food or other particles outside the cell. It like engulfs them and then brings them in. While the pinocytosis, I hope I'm saying this right.
the pinocytosis, we saw the first 3 letters were pen, so we're like a pinch.
Pinch.
So like they're
Yeah, they like take little portions in.
little portions.
And then the receptor mediated, it's exactly like it sounds. It uses the receptors to
To get the stuff to come in.
yes, it's just like a special case or a special form of the pinocytosis.
I think we should make note though that humans use receptor-mediated endocytosis.
That is
Yeah.
Yes.
that is an important.
That's a good
fact.
And they do this in order to take in cholesterol for their membranes, and as well as other steroids are taken in with the pinocytosis.
So I think we sort of thoroughly went through that chapter.
Yeah, I think chapter 7's good.
Let's move on to chapter.
No.
What do I want to do?
Let's do some cellular respiration.
Yeah, let's do cellular respiration. I think that's good.
What chapter?
So then we're moving on to
That's chapter 9.
So chapter 9 is cellular respiration and
Yes.
fermentation, if I'm not. Yes, and
Yes,
fermentation.
and fermentation.
And fermentation.
Well, I always forget.
I know. In my head, it's a part of the cycle.
Yeah, but it's not.
No.
Just get that out of your head.
Gosh.
Failure. Just kidding.
Sorry. Alright,
So-
kick us
So
off.
cellular respiration also kind of cellular respiration is works hand in hand with photosynthesis. It's almost the same
Cycle.
It's almost like the same cycle,
It's
but it's
backwards,
reversed. It's
yeah.
basically backwards. And so basically, what we're talking, the formula for cellular respiration is basically C6H12O6 plus 6O2, and it converts it to 6CO2 plus 6H2O plus ATP and heat, which basically means it's oxygen and sugar, and it turns it into carbon dioxide. Wait, you're right, carbon dioxide? No, it's not carbon.
Carbon dioxide.
Yeah,
Yes,
carbon
it's
dioxide.
carbon dioxide.
And then it turns it into heat, basically.
ATP
And glucose.
and
Glucose.
heat.
Yes.
It's and it's yeah, and there's also like the
So anyway, we can start with
This
For
all
us-
happens in the mitochondria.
yes. That is important.
Yes.
That is where it starts.
Yes. Whereas the photosynthesis occurs in the chromosomes of the.
Chromosomes.
Sorry. Oh my gosh. The chloroplast. The chromosomes. Yes. The chloroplast
The
within
chromosomes.
The chromosomes.
The
Oh my
chloroplast.
word.
That is an important distinction. Obviously, that's what sets us apart from plants. You don't have chloroplast. Fun
Yes.
fact.
Yes.
So
Also,
We don't have chromosomes.
we also don't take in our energy from the sun, as in the chloroplast taking their energy from light.
Yes.
well, we're getting into chapter.
But then again, we are getting to cellular respiration, so let's get on that.
so this is a catabolic process, right?
Yes.
Mhm.
It
Yeah.
is.
It breaks things down.
Yes.
Okay.
No cap.
We can start with the actual process. The first step is it occurs in the it's the first step is glycolysis, which
Which is in the cytosol.
which is in the cytosol, or what's the other name? It's just the inner cell membrane, right?
Not in the membrane, but in
But
the cell.
The outers.
Yeah, the outer bit. Yes.
Wait, no, I will find it.
No, it's in the cytosol.
I know it's in the cytosol, but there's another name for it. I'm pretty sure.
Is it?
Oh, well, point is, it's in the cytosol. That's a correct name at least. I don't know why.
Got him.
And so I guess we could go through really quickly the overview. Before we go into specific.
Go by the steps.
Okay, so your
I was
first
gonna-
step
Okay, go ahead. No, you're fine.
is the lysolysis, which it starts out with your glucose, and then it breaks it down into 2-pyruvate, 2-ATP, and 2-NADPH, or NADH. Yes. So, and that's a substrate level phosphorylation.
Well, it moves into the matrix, where the Krebs cycle occurs, and it goes around in the not the Krebs cycle. Well,
The citric acids.
the citric acid cycle. The Krebs cycle is
Yeah.
sorry. The Krebs cycle is photosynthesis. The citric acid cycle. It goes around once for every pyruvate.
No, it's the Calvin cycle. That's photosynthesis. It's called the Krebs cycle and the citric
yeah, and
acid
citric
cycle.
acid. Okay.
Yeah. I love
Yeah.
that. So.
I'm sorry, I'm
No, you're
confusing
good, Molly, you're good.
myself.
Step 3 of cellular respiration is oxidative phosphorylation.
Which is the electron transport chain, for other words.
Well, both of them, they're two different things, but they're in the same step.
Yes. And basically how that works is the NADH gets split up from NADH to NAD, and then it separates it from the H, so it becomes NAD plus H, which goes through an enzyme.
And
oh yeah, you're gonna say it.
it takes place in the cristae, which is the inner membrane of the mitochondria.
And the way we make our ATP, we use ATP synthase, and we use it was something acetylcholine. Well, the acetylcholine, and then it turns it into ATP, which basically makes it. You know,
I think
what's the? It's like the coenzyme A, right?
yes.
Yeah, and then it uses coenzyme A, which makes it.
Acetylcholine.
Yeah,
That's
that's how I remember it.
That's
yeah.
how you remember it.
Yeah.
Yeah, that's understandable.
I think the process that occurs in the electron transfer chain is pretty interesting. I remember we talked in tutorials about cytochrome complex, which I didn't
Mhm.
fully understand until you explained it. It was like taking off the H ion from NADH and FADH, and
That's it.
Thank you.
FADH2.
two, yes. And that was how it was being processed, and then it would go into the ATP synthase, which is a really cool structure, might I say. I can't believe it's like a turbine thing, like turns
Mhm.
when you. Anyway, I don't know why I thought that was
Never mind.
Wow, our cells can just do too much, bro.
too much.
Too much.
Um,
And so, yeah, that's basically cellular respiration.
I feel like we made it sound so much more simple than
Than
it
it actually is.
Yeah.
It's like-
complicated.
It's really complicated, but
Well, we could talk about what happens if you don't pass through. So, like glycolysis, if you don't complete that glycolysis, what happens is fermentation. Yes. So, lactic fermentation is I love an example that you give in class, like how when you work out and you like cramp and stuff. That's because the lactic acid.
oh, and fermentation.
He knows a lot about that.
Fermentation is whenever a process doesn't have oxygen. Whenever.
Yes, a lack of oxygen, and there are two. Well, never mind.
Okay, so there's two different phosphorylations that happen during cellular respiration. One is substrate level phosphorylation, which happens during glycolysis and the Krebs cycle. And the other one is oxidative phosphorylation, which is the last step, which is what changes like ATP to ADP, which is powered by redox reactions.
Sorry.
We should move on to. I guess that's a good way to like
I
describe
don't.
it. Or, like,
Let's connect it to photosynthesis really fast.
Nice.
Yeah, photosynthesis. That's what I'm talking about.
Oh, okay.
Yeah. Which is a
Photosynthesis.
good way to lead a photosynthesis, is what I was trying to say.
Yeah, photosynthesis occurs in autotrophs, so
Yeah.
organisms that create their own food, basically,
Yes.
which is generally mostly just plants, but it's some bacteria as well.
Mm-hmm.
This process starts off with photosystem 2 in the electron chain, and NADP turns into that goes into PlayStation 1, where it turns into
That's how we remember it. PlayStation 2, bro. PlayStation 2, Adam and I say, is the best gaming system. Therefore, it goes first. And that's basically how we remember photosynthesis.
Oh, that's
Fun
nice.
PlayStation
Fun
1.
facts, though. The reason why it's PS1 or PS1 or PlayStation 2 goes first is because it was discovered first, and then the second part was discovered second, and that's why it's reversed.
Never actually thought of that. Interesting.
Okay.
Wow.
Or I said it backwards.
Yeah,
but
yeah. We got what you
Yeah,
meant.
yeah,
We
sorry.
got
Yeah. So how about what goes after that? PS1?
well, NADP plus turns into NADPH, and then it goes into the Calvin cycle.
I guess like the equation. Oh, I was gonna give them like the equation for photosynthesis is kind of literally like kind of the opposite of cellular respiration. It's 6 CO2 plus 12 H2 plus light energy, and it makes CH12 O2 plus 6 O2 plus 6 H2O, which is basically carbon dioxide, water, and light, and it turns it into sugar, and oxygen, and water.
And how the Krebs cycle has the acetylcholine, the Calvin cycle has ribosome, and that's also what leads to your carbon fixation.
Another name for the Calvin cycle is the dark reaction. So that's kind of how it's split. The photosynthesis is split. Light reactions and dark reactions. Contrary to the name dark reactions, they usually happen during the day. So, fun fact.
So PS2 and PS1 both take place in the I don't know how to
Thylakoid.
thylakoids.
Yes, the thylakoids.
And then the Calvin cycle takes place in the spermata, which is the liquid stuff.
Inside it.
Yes. Okay. We basically, yeah. And like, so photosynthesis is like the exact reverse of like
If
cellular
you watch
respiration.
cellular respiration backwards, that'd be photosynthesis.
Photosynthesis.
It'd be photosynthesis.
Except for the chloroplast, then again, it is the
Except for- in a
mitochondria.
plant.
Yeah, and then
Then
and the difference of the enzymes as well. And then like there's a few like different because it's like the G3P instead of being the pyruvate, now it's the
G3P.
bracket CH2O bracket thing. So, yes.
Okay.
Anything else you want to add?
I mean, we could talk about the light.
Talk about like the stomata is on the bottom because it can't get wet, right?
It's like
Yeah, stuff can't get in it or else it'll clog up. Like when she, Miss Gaston, not she, Miss Gaston talked about how if your nose was upside down, like everything would get inside.
yummy.
That's
like
a
a
really good
nightmare.
color.
Yummy. I mean, why wouldn't you want that?
My friends like dirt from clogging it up,
'Cause
so
it's
it can
like-
release.
Red, green, and blue
Yes,
Yeah, it's
those
the
are
primary
two
colors.
make white.
Make way.
There are colors that are absorbed, and then the leftover color is the color that's reflected, which I personally did not
That's
know
insane.
at all.
Yeah, I know,
I-
that is super cool.
Yeah, I didn't realize that's what was happening, and that's why like the primary.
Well, your secondary colors are shown.
Yeah.
Interesting.
Yes.
Yes. Okay.
And chlorophyll A is the key light capturing pigment, and the accessory ones are the chlorophyll B and the how do you say it?
Carotenoids.
Carotenoids.
The
Yes.
more colors you have with these, the better. The chlorophyll A, which is what I found really interesting, was during the sunrise and the sunset. Like, it has the red for that, and then for the midday they have the purple. So I think that was really interesting on how.
So they get the most energy from the rise of the sun to the sunset.
Yes. So I thought that was really interesting.
From the sunrise, it's like reddish to the like sunset where it's like dark purple.
So basically, environment or
It adapted
habitat
to-
has a lot to do with
Yes.
what the plant looks like.
Yeah, it's really cool. So I guess like, would the environment impact what? No, I guess no. I'm stupid. Never mind.
And then basically to confirm the working, like Jose and them were saying about the sunlight or the sunset and the sunrise being the different, I guess, the waves on the light spectrum. You can there's actually called an action spectrum, and this theory, I guess, is basically tested whenever they shine different like wavelengths of the lights onto chloroplasts just to see basically the rate it's able to
Photosynthesis.
photosynthesize, kind of like similar to the lab that we did in class.
That's exactly what we did.
That
in
is
the lab.
that is definitely the lab where we tested the different UVs onto our spinach leaves. So yeah.
That's how some
Thank you.
some of the lights reacted faster than others. For example, I guess like for me, I had like the outside like light, like the actual sun
The
and
window?
the window, so we could see how like
The outside light.
the leaves did rise faster due because it was starting. What was it? Photosynthesis, right? It started photosynthesis, right? And it started floating up, while others were still waiting for theirs to float. While they were using like someone was using
like lamps and
yeah,
stuff.
Like a
lamp.
candle.
Whenever the
Yeah,
person
the
was using the
candles.
candles, we're like, that's not gonna do anything.
Yeah, like I'm pretty sure like the ones that used the red light didn't rise as fast as ours, so I think that could be a reason why.
Yes.
Yeah, and then like my group had the white light, and ours seemed to rise pretty fast.
Yeah.
So.
I think it also had to do with the baking soda that we put in. It used to
Oh
see
yeah.
So like it was typically the test one that rose had at least the most spinach pieces
Yeah.
of spinach.
Yeah,
that
that's what I
rose.
noticed when we
It's
looked
because
at
it was
them.
using the CO2 to go into the photosynthesis along with the light, and then that created oxygen.
Why did the
molecules,
cyanide
and
like
it
block it?
rose.
How did the cyanide block it?
The cyanide goes like attaches to the it goes through the mitochondria and like makes it not
It binds.
Binds to it.
Says hello.
the
receptors.
yes.
So has a later reaction to it. Like, well, like,
There's like no reaction because it
just
stops it stops it at the electron transport
Transfer
chain.
chain.
So
and
they're
for
just like
So the
plants,
very
it's like
beginning.
the beginning.
Yeah.
So for plants,
It's
it's
the
in the
very
very
beginning.
beginning of the chain, so it never starts.
So that's why whenever we went through our lab, that's why there was like all of the other stuff. There was like a build up until you got to the electron transport chain, and after that there was like such a limited number.
The reason why they floated, actually, Miss Gaston told me, was because it was releasing oxygen. I mean,
I literally just said that you have.
oh well. just a
Because
little
whenever you go through photosynthesis,
you release oxygen.
you release oxygen.
It's
So
one
of
of
course
them.
they would float, they're releasing oxygen.
Yeah, just a little, my 2 cents, if you will.
A little aggressiveness just to put it back in their mouth. A little positive reinforcement. Alright. I think we're good.
Yes.
Awesome. Thanks for
Thank
listening,
you.
Miss
Thank you.
Gaston.